Professor Angus Cameron is the second son of Alexander Cameron, who was a consultant at Norfolk and Norwich Hospital, and Elizabeth Cameron, who was a ward sister at the Middlesex Hospital. Those were the days. After education at Thorpe St Andrews Secondary Comprehensive School, Angus proceeded to study biochemistry at the University of Bath. Great location. Which included an eventful year working in Virginia Tech and St Jude's Children's Hospital. It was during this time that Angus fell in love with the laboratory and protein phosphorylation, resulting in his first paper. Angus then went to PhD studies on innate immune cell signalling at the University of Glasgow under the supervision of Janet Allen, publishing papers on the coupling of immunoglobulin receptors to intracellular signal transduction cascades. He also collaborated with actually my very good friend, who sadly I think passed, Alan Jardine, on novel mechanisms of action of statins on immune cell activation. He then moved to Cancer Research UK at the Lincoln's Inn field site to study protein kinase regulation of cancer cell biology with Peter Parker, who's here this evening. During this time, Angus developed a deep interest in protein kinase allostery, and a series of manuscripts revealing novel kinase activity followed. He established his own research group here at the Barts Cancer Institute at Queen Mary in 2013, focusing on the role of the Rho-regulated PKN kinases, which play a central role in mechanotransduction signaling in diverse cellular contexts, and which are now key emerging targets, which we're going to hear more about in a minute. So he also developed novel PKN targeting drugs in collaboration with the Dundee University, presumably that's the phosphorylation unit in Dundee. Angus is now Director of Graduate Studies for the Barts Cancer Institute, overseeing all of our postgraduate research degree programme, and teaches on a variety of undergraduate courses, including tissue pathology and personalised medicine. He's an enthusiastic supporter of patient and public outreach, which is very, very important in modern science, particularly working with schools and cancer patients. So Angus is married to Amy, and they have 3 children who are here this evening. Lola, Fraser, and Eddie. Welcome. So now I invite you, Angus, to give your inaugural lecture. Thank you.
Should we get a drink? No. Yeah. Thanks very much. Thanks for those very kind words, Mark, and thank you all for being here, especially my mum, who's made it, which I'm delighted about, and all my brothers, my children, and so many faces from the past and current stuff that I'm doing. So. Kinase families and friends, this is a bit of a play on words because I work on families of kinases, but of course this is actually as much about families and friends as it is about the science. And it's a bit difficult, where do you start with something like this? Well, I think the only place to start really is with my parents. And they came from extremely different backgrounds. So my dad grew up in a very Presbyterian Scottish coal mining village called Trinent. And his dad was a coal miner. In fact, as many coal miners do, he died when my dad was very young. My mum, on the other hand, came from Epping, from farming stock. And so really this was a matter of socialists meet conservatives. And it was really, they couldn't have been from more opposite sides. But what was amazing was being as Scotland is a socialist country, there's this thing called social mobility they have there, which is really weird. But apparently you can come from a village like that and still go to university. It used to happen apparently. So actually, Queen Mary is really brilliant for social mobility, so that's actually quite a good link. So he, when he went to school, he was basically given 3 career options, my dad. It was the clergy, the law, or medicine. Now he's an atheist, so that was out. He hated lawyers. And so therefore he would only really have one choice, which was medicine. So he went on to study medicine at Edinburgh. And my mum, on the other hand, she was probably expected just to marry a farmer and stay in Essex, but instead she was determined to do something valuable with her life. Not that that wouldn't be valuable. And sounds like a cushy number, really, but she instead went to become a nurse, and she studied and ended up at the Middlesex. And she was very good at this job, because she ended up in the sort of intensive care wards. Here's a couple of pictures of her next to some, well, it looks like sort of one of our fax machines, really, but here, this is a life support system of some kind, Mum? What is the machine? What is it? What is the machine?
What date?
What is this machine?
Well, it was a defibrillator and a ECG, and
Oh.
but you couldn't take temperatures to anything.
Yeah. So she, does anyone know who that is?
There's my old boss.
It's her old boss. So she was so good at nursing, she ended up the ward sister on Sir Thomas Holmes Sellers' ward, and he was one of the absolute pioneers of open heart surgery right at the beginning. So she was really good at her job, and you know, I'm very proud of her and what my dad achieved as well. And so my dad went on to become, after a stint over at Christmas Island watching nuclear tests as a medical officer, he went to the Norfolk and Norwich to become a consultant there. And the clicker stopped working, here we go. And so we grew up in Norfolk. So we moved out there when I was about 2 years old, and then my two other brothers, Hamish and Dougal, followed quite shortly after. But there's a reason why, you know, I was born in North London, and that's why I'm an Arsenal supporter, and my brothers, they were born in Norwich, and that's why they're Norwich supporters. So just for a bit of context. So here's a picture of the four of us back in Norfolk. And as you can see, we've got our names on our T-shirts, and it's Duncan, Angus, Hamish, and Dougal. And I think this is probably slight overcompensation for a Scotsman who's moved out of Scotland. But you know, we didn't get that bullied. It was fine. And here's me doing an early experiment with some mouth pipetting down the bottom here, and covered in mud, because basically we spent the whole of our life just crawling around in the mud in the garden. And my mum particularly likes gardening. There's a nice picture of her gardening up there. And I was an incredibly inquisitive child, and I used to just badger her relentlessly with question after question after question. I'd be going, why is the grass green? Would it still work if it was blue? Can you eat worms? And she would be going in the garden going, uh-huh, uh-huh, uh-huh. And I was like, Mum, what does uh-huh mean? And she went, well, sometimes it means yes, sometimes it means no. I said, no, it doesn't. It means you're not listening to a word I'm saying. She's listening now. Right, so early years, we'll skip a bit. I promised my lab I'd put in a picture of me with long hair. And there's one at the top right there. I had lovely auburn locks when I was younger, which my dad hated and really wanted me to cut off, but, and I have now since cut off. And then there's a picture of me with some of my brothers at school, and I just wanted to put in, this is a picture of my school, Thorpe St Andrews Secondary Comprehensive School. It's a wonderful school. My mum was a governor there for some 30 years. And I put in 3 teachers here who were really inspirational at that school. One is Owen Hunt, who took us on all these crazy expeditions to Scotland, to Wales, to climb the mountains, and even a month in Iceland. And that really lit up my, you know, my absolute love for the outdoors and the mountains. Second was Richard White, I've got one of Richard White's sons here. He never actually taught me, but he was an English teacher, and he used to run all the school plays and all the music, and so he ignited a huge love of that in me, which has been squeezed out by science now, of course. And then, the last but not least, Dr. Claude Scott, here with the white hair, he was the headmaster of our school, and he was the chemistry teacher. And, you know, my favourite subjects were chemistry and physics. I mean, in fact, I turned up at secondary school. I was really, really short. I think it was called Failure to Thrive. And I was ginger, and I was really good at physics and maths. I mean, it's amazing I'm here, to be honest. I mean, I would have bullied me. But anyway, I eventually grew to almost normal height. And, but he was a really inspirational teacher, and he really set me on my path to studying science. He would have made it today, but it was a little bit of a tricky journey. I couldn't handle both him and Mum, basically. It was one or the other, and Mum won. Okay. So my heroic brothers, right, here they all are. So this is Dougal on the left with his chainsaw. His favourite place is chainsawing in the woods. So if he could do that still as his full-time job, I know he would, but now he has to teach maths instead. And then Hamish in the middle, full hero, RNLI. So he lives down on the Devon coast and works with the council, but this is what he really loves doing. And then there's Duncan, my brother there, and he's an artist. And this is him winning the Ruskin Prize just a few months ago, which was an amazing evening that I was at. And so Mum's got lots of reasons to be very proud of them. But, you know, my mum and dad, really, this is just to highlight that mum and dad basically let us do what we want. They say, you can do exactly what you want, and we all do extremely different things. Duncan and Dougal dabbled in medicine, and here's a couple of pictures of them in their training, but somehow they didn't inspire much, you know, trust in the patients, so they moved on to different things. Hamish just likes being a patient, and here's some pictures of him being helicopter rescued and then wheeled out, which he's done more than once, actually. He's quite good at it. So I went to Bath University to study biochemistry. Now, you know, I love chemistry, I love biology, but I actually have to say I found the biochemistry degree a bit, can I say boring? It was a bit boring. And it was just all this, you know, sort of sugar molecule conformations and plant biochemistry at 9 in the morning, and you're like, you know, having just grown to normal height, I was just trying to get a girlfriend. I mean, you know, it's like it just was not interesting enough. So, but very lucky, the Bath University courses are 4 years long, and they have these year placements. And I went to the States for a year. I was really, really lucky. I mean, because it was really formative. And when I was in the States, I worked for this great guy, Peter Kinase. And he said, I really like this quote, he said, I'd argue that curiosity, observation, and imagination all rank before intellectual prowess. And I thought, hope. And, but his lab was absolutely amazing. So we worked on sulfolobus sulfataricus, this archaeon, it's an archaebacteria. And we were purifying enzymes out of archaebacteria that grow in hot vents, and to try and develop enzymes that work in different circumstances. And so basically we had these huge, great big vats of this stinking yellow broth, and we stank the whole building out. Everybody hated us, but we absolutely loved it. And then we were looking for phosphatases and kinases, and so we were just tipping P32 around everywhere. I mean, it was complete. I mean, honestly, Gavin would have died. I mean, you would have been like, this is... And so we're tipping it, but it was just brilliant. And I just completely fell in love with being in the lab. And, you know, that really what sparked my desire to do science. And I'm going to show you in a few different labs, just 1 or 2 experiments. But this was, so there's me with long hair in the dark. I found it in a photo album, but it's not very well reproduced. But that was, so I did this experiment where, you know, back in the day when you did science, you'd make these huge cell lysates and tip them down big columns and get lots of fractions of proteins and do biochemical assays on the fractions and look for different activities. And so we were trying to identify phosphatases, and we had this radioactive phosphatase assay, and here it is. But we had this gel of the purified protein, but of course there are a number of different proteins in here, and we didn't know which one actually had the activity. So I suggested to the boss, well, why don't we just cut the gel up, squidge it all up, and do some phosphatase activities in the fractions? And he said, yeah, it'll never work, never work. But it did work. It worked beautifully well and showed that this protein here was indeed the fraction in this sample that had the phosphatase activity. And this was, you know, before genomics, and you then get a bit of N-terminal protein sequence, make a DNA probe, a degenerate probe, and pull out the cDNA and clone the gene. And I only did the purification bit. I didn't do the other bit. But I got on paper, and it was really, really exciting. And my journey with protein phosphorylation began. But I hear you all say, what is a phosphatase? What is a phosphatase? What does that do? What does it even mean? Right, so I'm going to give a short lesson on protein phosphorylation. So this is what they normally show. You've got a protein here. A kinase puts a phosphate on the protein. That causes the protein to change behavior. You can have a phosphatase, which can take the phosphate off. But of course, I think we need some help with this. So who are these people, Fraser, Edie? Who's that up there? Is that Ellie? It's Ellie, isn't it? That's right. Well, so Ellie's going to be our kinase for the day. And Ellie's holding on to ATP, which is the apple. And then we've got Shelley, who's a protein who's about to get phosphorylated. And so Ellie comes along and phosphorylates Shelley. Now, I know, Peter, they don't take the whole ATP, but yeah, it's just the phosphate. For now, we'll take it as that's the phosphate. It's a bit shifty. He'll be telling me off afterwards. Oh, I feel like I'm a Viver again. Oh, God, I'm sorry. More on that in a minute. So then when she's got the phosphate, off she goes to see her new friends, because so it's changed her behaviour. But who's this? Oh, my goodness, here comes Tigery. Tigery's off because Tigery likes apples, and munch, munch, munch. Off we get. Tigery is a phosphatase. So he's removing the phosphate group, and now she's hungry again, so she has to go back to see Ellie the kinase. So there we have, instead of this, we've got protein kinases, which can be elephants, there can be a few of them in the talk, and we've got phosphatases, tigers taking them off. So there we go. That should be the next textbook. There we go. Okay, so I just helped discover a new tiger. How very exciting, my own tiger. Right, of course it's more complicated. So now in steps Lola, my other daughter, who's crocheted all of these animals you see, all of these animals. She crocheted. She just developed a skill overnight. And she makes all these animals. I should put up a website for where she's selling them, actually. It's quite a good audience to get us some sales. But so in human cells, there are over 500 different kinds of elephants. And basically, these phosphorylate networks of proteins, and you get these network of communication in the cell that passes messages from the outside of the cell to the inside of the cell to tell the cell how to behave, to grow, to move, and what have you. Now, the advantage of these is that there's a really useful thing about kinases, which is you can make drugs that bind to them, which means, and we're going to have the drug here is going to be a toffee apple. So the drug here can come in and stick to the kinase and stop it working. And now a huge number of drugs we have for changing the biology of cells are protein kinase inhibitors. And a huge number of drugs against, in cancer are protein kinase inhibitors. So this is a huge area, and it's about identifying which kinases do we want to inhibit and where. So you can actually, potentially make different inhibitors for all of the 500 different kinases to make, to basically switch off behaviors you don't want. Okay, so that meant that I finished my time in Bath, passed my degree, which is good, and went up to Glasgow to do my PhD. And Glasgow's, I don't know how many people know Glasgow well, but this is just an amazingly wonderful city, and I had just the best of time there. But as with almost all the places I've worked, I always work in a really brutalist 1970s building that's falling down with a beautiful view. And I did that here. And I continued this throughout the whole of my career. I've never worked in a new build, ever. So here's me at the beautiful Davidson building where I met Tommy. Where's Tommy? Yeah, Tommy, sorry. Yeah, I've got my glasses on. So this is the Davidson building, the biochemistry building. And these are the people I work with. Janet, who I was going to be here, but I don't think has made it. Then I also worked with Maggie when Janet went off to Pfizer in France. And I also struck up a great relationship with Alan Jardine, and who worked at the Western Infirmary. And between us, we published a few papers, and it was a really exciting time. And I absolutely, fell in love further with protein phosphorylation. So I'm going to say a little bit, just one little experiment about what we did there. So I used to work on Fc gamma receptor signaling. So these are the receptors that bind to IgG, antibodies. So when you get an antibody attack against a bacteria or a virus or something, the other end of the antibody that gets the immune cells to actually do something binds to Fc receptors. And we were very interested in how these receptors switch their signaling molecules to change the signaling pathways they engage. So I think we should bring in some friends again. So basically, we had a situation where when we differentiated cells into phagocytic macrophages, this is Fc gamma R1. You can get a signal that goes from IgG to activate this protein phospholipase D. But when you differentiate the cells to macrophages, what happens is this other receptor comes along and sneakily steals the mate. And now the signals go down via this other signaling pathway. But now it's switched to an IgA response rather than IgG. And I remember developing this blot. I remember when it came out going, that would be an interesting theory, wouldn't it? And the reason I put this experiment up isn't because it's the best experiment I ever did. It's just I completely remember the moment when it came off the thing and I go, that's right, that's it, that's the answer. And so, it's those ones that, it's that love of science that's, kept me going all these years. But it wasn't the only thing I did in Glasgow. I was completely obsessed with rock climbing. And Chris down at the front here, he's still climbing. That's him, like kind of with his top off here, in a balmy Glasgow evening. And this is all out at a place called Dumbarton Rock, which is about half an hour's drive out of Glasgow. And to many people, it would be a godforsaken place because there's smashed glass everywhere. There's graffiti on all the rocks. It's dark and rainy a lot of the time, but it was just a really amazing place. And it's got crazy, huge boulders to climb on. It was a very exciting place to go. And yeah, I loved it. And I met friends who I still see. I don't climb with them so much these days, but it's maybe London. Okay, so, but I did produce a thesis, and for anyone in here who, hands up any PhD students? Within the deadline. Just, keeping my professional hat on there. So the thesis, I dedicated the thesis, this is the bit where I might struggle. I dedicated the thesis to my dad because I never thought he'd make it to me finishing my PhD. When we were young, he suffered a lot from heart problems, and frankly, I thought we were going to lose him before we'd even got to secondary school. But somehow through amazing heart surgery and bypasses and drugs, he survived, and I managed to dedicate my thesis to him, and I think I even raised a tear, and that was one of the proudest moments of my life, and along with having my children, getting my mum to come and sit here at this lecture is in that same space. So it also led to my first conversation with Peter Parker up here, because Peter was my external examiner on my Viva. And so he duly came up to Glasgow to do my Viva, and it was some of the hardest 2.5 hours of my life, because the questions were so difficult. He kept asking me like what the IC50s of this inhibitor are of that one, what the KI for this protein, and I was like, I don't know. And he pulling out, why on earth would anyone use this at 250 micromolar? And I was like, well, it probably is a little bit high. So he sort of left this with me, sort of, with him thinking, who is this cowboy? And me thinking, but I thought I might learn something. And very kindly, he obviously, saw that I needed to be corrected a little bit more, and he let me come down and work for him in his lab. And so here's the next really brutalist, ugly building that's now no longer there, looking out onto a really beautiful square at the LRI. Amazing memories of that place. It was just a brilliant lab to work in. It was just great fun. And between all the fun, we did actually look at quite a lot of interesting protein kinases. I'm going to skip through this because we haven't got very much time, but we published on various kinases involved in growth factor receptor signaling, which are the signaling pathways that tend to go wrong in cancer because they tell the cells to grow when they shouldn't. So these are obviously potentially drug targets. So I'm going to, I spent the majority of my time working on this family, so this is why my talk's kinase families, this protein kinase C family. And there are a lot of members in this family, and I've worked on quite a few of them. So I'm just going to tell you one story about PKC alpha because it has quite an interesting ending. So when I started with Peter, one of the first projects I worked on was the role of PKC alpha in glioma growth. And we'd noted that actually, if you look in these 3 glioma cell lines, some of you will be thinking, where's the loading blot? I mean, where is the loading? Anyway, there is a loading blot, but they've got loads and loads of PKC alpha. And in this experiment here, we show that if you knock that PKC alpha down, if you get rid of the protein, enough of it, then the cells die. We also noted that actually, if you take a protein kinase, an Ellie, there's Ellie, and you make a mutant that now can't bind ATP, so it's not active, that also kills cells. So this gave us some confidence that actually, this is going to be a drug target. We should be able to target this with drugs and stop the glioma cells growing. But the reality is, the drugs just don't work at all. I mean, you can inhibit PKC as much as you like, it just doesn't do anything. So we had this paradox. It's like, why is genetically killing the kinase activity different from inhibiting the protein? So, why is that different? And it turns out that it's a very, this is cutting an awful lot of work very short, but it turns out it's because when you have the wild type kinase, this allows cells to grow. But if you stop it binding ATP, it causes cells to die. But if you re-engineer the way you kill the activity of the kinase, so it can hold on to the ATP, it can hold on to the apple, but it can't let it go, then that causes cells to grow. And that says it has to do with the conformation that the kinase is in when it's bound to the ATP. And it hasn't got to do with the lack of activity. And you can go on to show that actually an inhibitor in the pocket actually stabilizes the pro-growth isoform of that kinase. So of course those inhibitors don't work. And this, is a rather profound, it sounds quite esoteric, but it's quite a profound observation about the way we interpret some of these experiments. So I published these papers with Peter in 2008, 2009. I'm going to jump now 10 years forward. So I've set up my own lab years later, and out of the blue, these papers start appearing, where this rare mutation is appearing in this subset of glioma patients, chordoid gliomas, where there's this mutation in the exact same residue that we had used to disable the ability of PKC to let go of ATP. And it turns out that in nature, this D463H mutant is indeed a kinase-dead ATP-locked conformer that drives glioma growth. And so we restruck up some collaboration to chase this down. And between Veronique with Peter and Jack and Joe in my lab, we sort of chased down this novel mechanism. And what we essentially showed, I'm jumping through this because I haven't got very much time for wine. What we essentially showed was that when you have this mutant, it's trapped in this ATP-bound conformer, and that alone is enough to drive the oncogenic signal. And if you introduce the mutation, even heterozygous, into a murine model, that alone, on its own, just as a single heterozygous point mutation, drives chondrosarcomas in the mice. So they don't get glioma, but they don't live long enough to get glioma. It drives a chondrosarcoma. So it's an oncogenic mutant, but there aren't that many mutations where you have a heterozygous endogenous point mutation driving a tumor, but that is one. And actually, this is just looking at proteins. This particular mutant pulls down a load of proteins that don't bind to the wild type or anything else. And there are some really fascinating hits in the interactome, including a number of epigenetic modulators. And we think that's the mechanism. And Neil, who's also in the audience here, and I have got a PhD student starting on this in September, which is very exciting. OK, so when locked in this conformer, these gets all these new friends who like to hang out. You see some of your other friends there? OK. So this really nails down PKC alpha as a proto-oncogene, which is kind of a bit of a grey area. There are gain-of-function mutations. Kinase inactivating gain-of-function. Weird. There are also, the mutation alone causes cancer in a mammalian model. It's amplified in settings. There are fusions. But classical inhibitors are probably not going to be effective. So people who meet on the way had great fun in the Pip Parker lab. But I did also meet someone rather special while working there. And this is I cut a dashing figure back then, So this is me chatting her up. How that worked, which I'll come. Yeah, there's a story about that actually coming up, I think. And before we left the Parker lab, we had our first child, Lola, who is now very nearly as tall as I am, and this is Peter holding her before she was 1. So the last few years, I'm going to jump through this very quickly to get to the start of my lab, but I worked on this family of protein kinases, PKM, which are related to the PKC family, and spent years developing in vivo models, and it's those in vivo models that I brought to and work primarily on here. So this is another group of elephants, and this was a big team effort, and I've put some of the people there who were important to it. But what we noted was that PKN2 is the most interesting kinase in the world. And, everyone knows that I work on PKN2. It's like I remember I wrote a grant once, and we sent it round to colleagues, and they go, you lost me in this grant at the first 4 letters. So I've stopped putting that as the first word, sentence 2 or 3. But so one of the things we noticed is if you make knockouts to these of the PKN2 gene, you get really, really profound breakdown during development, and the embryos die very young. They have a really fascinating vascular phenotype, and this is what caused me to meet Kebs, because Peter knew Kebs and said, "Oh, vasculature, go and speak to Kebs." So I came and I had a brilliant meeting with Kebs, where I just went, "Oh, it could be this, it could be that." And she went, "It could be this, it could be that." And along with the science, she pointed out that there was a recruitment drive going on at Barts. They were looking for new lecturers, and I should apply. And so actually, I came over for the vasculature and left with a job, more or less. I mean, it wasn't quite what I expected. There were a few steps in between. And so I started the BCI, and here's a picture of us all celebrating just a few days after I got my lectureship with Nick Lemoyne phoned me while I was outside the Seven Compasses pub near the LRI and told me I'd passed the interview. And, I was absolutely delighted. And this then started the period, the starting up a lab. Everyone who's done it knows it's pretty stressful. But Kebs had this nickname for me. She called me Stressed Eric, because I'd come in and this vein on my head would be just go bang, bang, bang. I'd go, got to write grants, got to write grants. And that went on for probably she's like, has it stopped? Yeah. Yeah, a little bit less. So and during this time, I put a few people up here who were really, who really helped along the journey. The 2 Nicks here on the left, Nick Rice and Nick Lemoyne, And then Jude gets a special mention because during the first couple of weeks here, we had these whiteboard meetings where basically you put your ideas up and then people sort of shoot, argue with them about. And he absolutely ripped me to pieces. He just literally just told me, rubbish, you're never going to fund it, rubbish, rubbish, never. And I was just like, oh, God, this is awful. What have I done? I had a nice job in Peter's lab. So, I was terrified of him. And then, I had this CRUK drug interview that I had to go to. And I was like, you do some mock interviews. Never, you've got to have Jude on your mock panel because he'll ask difficult questions, but they'll really help you for the interview. And so we do this interview, and it was about getting some money for making PKN2 drugs. In fact, you and I did it together, Louise. We didn't get the money. And we sat in this mock interview, and he goes, so Angus, are the other drugs going to work? And I went, well, I mean, they probably will. I don't think they'll be. And he just stopped me and went, Angus, I'm amazed you've got a wife. I have. Look, there's one there today. And since that, I fell in love with him after that. It was great. And he really went out of his way to really help me choose between my projects which ones people would actually give me money for. And that's probably the hardest lesson to learn. But the advice he gave and the projects I chased down, like the Bowel project, I'll say a little bit about in a minute, if I don't know what time we're at. I'm doing okay. He told me that one will get funded. That one very interesting, but just no, a whole panel's never going to give you money for that, and that was really useful. And then also special thanks to Delphine, who can't be here today. Unfortunately, she got a bad back, but she was really hoping to be here. But she, she's kept this institute ticking over for the whole of the time here, but's recently retired. And John, who's my centre lead and a constant support for my work, and also to Nixon, our new director, who's just here. So thanks to all these people. So these were the 7 ECRs. We all started at the same time. And is Trevor here? No, he maybe didn't make it. 6 of us still work, actually, in the Institute. Trevor's gone on to work at the ICR. But all of us remain in science. So we all survived that initiation in those difficult years. And we all we all fight on to live another day. And, in science, if you're still doing it, you're winning. That's my sort of view. Before I just present some of the work we're doing in my lab at the moment, I want to say thanks to the people who did it. These are all my wonderful people in my lab. My first PhD students are up here. Lots of people from my current lab. They're the people who do all the experiments now. I really miss it sometimes. And in fact, I think I'll show a couple of examples of me getting back into the lab, to the slight horror of some people. But so here we are celebrating various events, including people passing their vivas. And maybe a special shout to Hazem, who's got his viva on Wednesday. So good luck to him on that. So we had this vascular defect in embryos. So that's a bit of a weird one, but it's not very cancery. And I'm now suddenly find myself working in a cancer institute. I think I'm going to have to do something about cancer here, aren't I? But what we worked out in the embryo in the end was it didn't have actually anything to do with the vasculature, the reason these embryos went awry. It was because actually all of the fibroblasts, and I'll explain what those are in the middle, failed to expand properly in the mesoderm, and also fibroblasts fail to migrate from the neural crest. So you get this failure of these fibroblast cells. Now, fibroblast cells are cells that grow in tissues that give tissue structure. And without that structural component, they just fall apart. And cancers are no different to that. And in fact, cancers, they need fibroblasts to support the extracellular matrix that allows the cancer to grow. And so we surmised that if PKN2 had this role in the developing embryo, perhaps it would also do that in cancer-associated fibroblasts. So we looked at there's a whole load of people. It's a great place to work on pancreatic cancer. There's a huge number of people who work on that disease here. And this is really sort of the archetypal fibrotic tumour. So the tumours are really fibrous, and they're really solid. And that's because they've got lots of fibroblasts in them making lots of matrix. And this makes them sort of impregnable and dense and very difficult to treat. And so we were hoping that actually by targeting PKN2 in those fibroblasts, we might change the nature of those tumours. And I'm going to skip through this quite quickly, but there's a really exciting result. And again, I'm just putting out results where I saw them and I'm like, wow, that's brilliant. And so here what we've got here is we've got cancer cells growing on a protein matrix. So this is a protein matrix, and the cancer cells are all growing neatly on top of it. It's like they don't even know they're cancer cells. But you stick in some fibroblasts in this middle panel, and they go absolutely mental. They grow much more, they invade into the tissue, and they become really aggressive. And this is because the cancer cells and the fibroblasts are talking to each other to drive this invasive process. But then if we target PKN2 in those cells, then we can revert it almost back to the state without the fibroblast in it. And this is the same, but with red cells for cancer and green cells for fibroblasts. And Haslam's Viveron, to cut a very long story short, this is because we found out that PKN2 regulates this critical pathway called the mechanotransduction pathway, so it's like mechanical stimulation of signaling. And when you target PKN2, you uncouple YAP and TAZ signaling to drive transcription. And this is really exciting because not only is that important in fibroblasts, that pathway is a really important oncogenic pathway in lots of settings. And there aren't many ways in which you can design drug strategies to target that pathway. So I'm going to skip past that. So Haslam's Viveron on Wednesdays to cover the fact that we've now translated that to look at a whole series of primary cancer-associated fibroblasts. So these are fibroblasts taken from human patients. And in these cells, it requires both PKM1 and PKM2, but it's the same pathway, and it's completely conserved across all of these different models. And this is a little bit of bioinformatics showing that you get the myofibroblast signature gets knocked down in all of these different fibroblast lines, and they in fact become much more inflammatory. And that's going to be really important potentially because what we're trying to do is make those tumours more, to improve the immune content of these tumours. And this just shows this specific pathway is always knocked down when you target PKM1 and 2. And rather nicely, if you make a signature from a stromal signature from those cancer-associated fibroblasts, it predicts that if we were to target PKN2 in those cells, the patients would do better. So we're hoping to develop this as a part of a combination strategy. But he has his Viver on Wednesday, so wish him luck. So I'm going to skip past that. So, drugs against this pathway would be really, really useful. So, but we don't have any drugs. So this is why I went to, we linked up with Dundee to try and make some drugs. So we did some big screening and a lot of medicinal chemistry. And a lot of this was conducted by Josie Williams, one of my PhD students. Is she maybe here? Maybe not. She isn't here, but it was a valiant effort. And we did indeed develop some very potent drugs, some toffee apples for these protein kinases. And in these three of our drug series, PKN2 is by far the most potently inhibited. And we're currently getting these into in vivo models, which is being done by Joe. So watch this space. But are they going to work to suppress this pathway? 20 minutes. I promise we'll be on time for the wine. So we've made these really good reporters that report on the activation of YAP-TAZ, and this was done by Munira, who's here from Malaysia. Give us a wave. Ooh. And she made these great reporter lines which make an RFP signal when the YAP-TAZ pathway is on, and you can so then we can use that as a platform to look for inhibitors. And rather beautifully, our inhibitors really nicely dose-dependently inhibit this pathway. And we're going to get these into in vivo models as soon as we can. In fact, we've dosed them in, and hopefully that's something I'm going to be reporting on over the next few years. So, why do we all do this? I put this slide in because in the middle of my time in here while setting up my lab, within a year, I lost 3 of my very close friends, all of them to cancer. Andrew here on the left to glioblastoma. He was a friend of mine. He was in my brother's year at school, and he was my flatmate right up to the point where I moved in with Amy. Nick, one of my oldest friends from university. Ed here is a very close friend as well. And he had a long battle with a carcinoid tumour. And in fact, it was Sophie, Richard's wife, looked after him amazingly well. And then Kev, one of our core climbing team, and a very close friend of Chris's and ours. And so, I put this in because I can't, at the time that this happened, I was like, this is all too much, working on this and having, but actually, when I've got a slightly longer view, it just reminds us why we do it and why it's an endeavour that's worth pushing forward with. So I tried to do something positive. It's a bit of light relief. So it's a marathon running. And, so through the time that that was happening, I decided to raise some money for brain tumours. Loads of help from the Institute, loads of help from colleagues. Everyone was very supportive. But it didn't quite go to plan. Because in the final 50 meters, something went wrong. Now, you know, in the era of AI image analysis, I don't think you probably need that to see what's gone wrong here. I think there might be something there where the top of the bone isn't attached to the bottom of the bone. So God knows how, but I managed to snap my femur in half during that marathon. And I didn't, and this was in front of the gallery at the finish line. I mean, literally, you could, you know, people were saying, should I crawl to the finish line? And I was like, that's very advisable. I'm glad I didn't, actually. I might not have, yeah, it might have gone badly. But anyway, they pinned it back together with this great big bit of metal, and I'm back running again, so it's sort of fine. And that's me back with my family. But, you know, really what happened through this period was, you know, amazing support from my colleagues. Hemant came and saw me at the hospital, and he goes, can I check your blood test results? And he said, and he came back, and I was thinking, God, what is he going to say? And he came back, and he looked, he said, no, you're fine, you just broke your leg. More positive story, just to end on a positive marathon story. This is my wife and us 4 boys running for the British Heart Foundation. And I don't know which is worse, running in an ankle-length velveteen dress for a marathon, or running in the armour, and I think there's still a debate about that. But I put this in because, you know, to be honest, I couldn't have done any of this without my wife, because when I was like spending all of that time writing those grants and, you know, putting my hair out with stress, and we've got 3 young kids, and then I go and break my leg. I mean, come on. I mean, you know, how could I be less helpful? And so I want to say thank you to him. Yeah, she's really been an absolute rock, and you know, I'm just extremely lucky, and you know, my kids, well, they're great. Okay, so full circle. This is getting emotional. Right, so full circle. Here's some early A. Cameron papers. You sit there thinking, God, Angus doesn't look that old, does he? You know, from 1973, I can't be right. So these are papers from my dad, because he was a general surgeon, but he spent most of his life working on bowel tumours. And on patients with Crohn's disease and ulcerative colitis. So the last bit of science I'm going to tell you about is just something we've been doing that touches on that topic, because it really closes the Cameron loop. I quite like this. So here's a picture of him looking very stern in the middle, back at the Middlesex Hospital. And I think I've got, with regard to colon cancer resection, optimism is frequently rewarded with this condition. I like that. That's good. That's a good one. And then, and you know, the best chance of curing colon cancer is to cut it out. And I think, unfortunately, it's probably still true. But we're sort of making inroads. So here, you know, when we were doing the work on the pancreatic cancer model, we noted actually that the mice often lost weight in the knockout groups if they were under stress. And so we did this experiment where you give an inflammatory stress to either the wild type or PKN-targeted mice. And the PKN-targeted mice lose weight dramatically more. They have more colitis in their bowel. They're super sensitive to developing colitis and Crohn's-like disease, and they survive less. And what's more, they go on to develop loads more tumours because of the inflammation in the bowel. And we think this is just because of the inflammation in the bowel to cut a long story short. So they get more tumours, more of them, bigger tumours. And the number of tumours they get directly correlates with how much disease activity they have with regard to the colitis. And what Jack's gone on to show is that this is because PKN2 acts as a tumour suppressor by suppressing inflammation, and it does so because it's a really critical player in the regulation of tight junctions. So here we've got some cancer cells grown on a plate, and we've stained for all the intracellular junctions, and we can then genetically knock out PKN2 and all of these junctions disappear. And we've done that in a number of different models. I don't think I'm going to have to, I think I'm sort of running out of time probably. But so this shows that if you look in the bowels, that actually you get loads less ZO1, which is this critical junctional protein in the bowels. And you can then also see in these bottom panels here that the epithelium, that's the lining of the bowel, is now all broken up and becoming damaged. And what's really nice, this is just bioinformatics, what this piece of data shows is if you make a transcriptional signature from knockout organoids, so where we've targeted PKN2, the transcriptional signatures really closely mimic what happens in human colitis. So we've developed a new colitis model, which we think is going to be very useful. So I'm going to skip through this too much, just to say that I got back in the lab. Here's me back in the lab running the radioactive assays, which is my happy place. And my lab all said to me, they said, you know, I don't think I've ever seen you so happy, like pouring the stuff around everywhere. But that could also mean that I'm just miserable the rest of the time. I'm not quite sure how to take it. But there's me in my happy place. And basically, you know, we're now trying to work out the exact mechanism and all the different proteins in the junctions, the PKN2 phosphorylates. And we've identified, I think, probably quite a handful of sites that are responsible. And this is out on bioarchives. We're just trying to get someone to actually publish it. So, finally, I'm just going to wrap up with a couple of other things that I do. I mentioned in my bio, we talked a little bit about outreach. I've been really lucky to have colleagues, particularly Louise, Richard, John, Adrienne, hello, who have invited me and allowed me to take a really big part of this over the last few years. And so I work with a few different outreach groups. Voice, which is a patient advocate group where cancer patients and patient advocates come to the university and they spend a week, fully intensive week with lectures and lab work where they do all the kind of experiments that we do ourselves and get a real idea of how to do that. And, you know, I put this picture in because this is what science is supposed to do. This is when we were running a PCR reaction to look for an oncogene. And the experiments worked, and that expression sums it up. That's why I do science, for that moment. And the experiments that I've shoved in earlier are those moments where I've gone, look at that, it's amazing. Because I don't think any other job really has something as exciting as that. So all these people who were great with that. I also work with the STARS program, which is something that John set up, which is working with school students from low science uptake areas, and it's just really rewarding. And it's something the BCI should be proud of. Very quickly, it was funded by these people. That's enough of that. No funders in here, I hope. Well, yes, yes, yeah. Is the MRC up there? Because they just stopped. More importantly, amazing colleagues. You know, over here on the left, I've got tumour biology, which is my centre, but just loads of colleagues throughout the whole institute. And I've shoved in, you know, I'm bound to have missed people off here. But you know, it's the BCI sets itself apart because of the collegiality here. Everyone works with each other. The university as a whole is so supportive, and you know, it's great to see colleagues from all over the university. It's you know, it's a very friendly place to be and a lovely campus to work on. And yes, which is why I stayed. And then thanks to all my helpers in the talks. So here we are as Tigery, the Ellies, and all of these other ones that you made, Lola. And then finally to my mum. I've got another. My mum is a superstar. She's 86 now, and she's made it to London, which is very brave. Well done. And she's a great survivor. My mum. She can bounce back from anything. I could tell some stories. I will do over some glasses of wine, but I just could not be more delighted to have her here and to be able to dedicate this talk to you, Mum. And with that, I'm going to end my talk.
Well, I think that reaction from the audience says more than I can about what you've just done. So it was a wonderful personal description of a whirlwind journey through science and your life. And I agree, what great parents you've had, letting you run wild across the countryside. I do agree that perhaps the number of Scottish names amongst your siblings is a little over the top, but you know what, Angus, you can take a Scot out of Scotland, but you can't take the Scots out of a Scotsman. I'll be arranging an urgent appointment with the radiation safety officer after some of the images you showed there. But what an inspirational speaker you are. You managed to animate kinases and phosphatases with animals and fruit. By the way, what's the T-Rex for? Oh, okay. Thanks. Thanks. I agree with you that Glasgow is an amazing place and that Alan Jardine was an inspired choice of a collaborator. But I did wonder about the wisdom of asking PhD students in the middle of your talk, as you are now the Graduate Studies Director, if they had made their written deadline. Could be awkward. Peter Parker was an inspired choice at Lincoln's Inn Field for the next phase. By the way, I think he asks everybody about 250 micromolar. Once spent 2 days in New York with him, and we had some very unusual conversations in which I had no clue what I was talking about, so you're not alone. He is a brilliant mind. Now, you mentioned Professor Sir Nick Wright, who was the head of medical school, one of my forerunners. Now, he managed to pull off a tank top, but I have to tell you, guys, it's you or kids, you've got to, if that stripy tank top still exists, send it to the charity tonight. It must never be seen on your dad again. People who give you a tough time in preparation, like Jude, are the people who help you get through the experience. It's better to have a tough time amongst friends than it is to appear in a room where someone might give you some money and find out what a tough time really looks like. Your choice of Dundee as a phosphorylation centre for your new potential inhibitors, I think, is a great choice. And the dose dependency that you showed can give us all hope there might be something in that for the future. And I think the other thing you've demonstrated is you have indefatigable determination to carry on, not just with a broken leg, but actually in every aspect of your science. And that's why we're delighted that you found a great home here at BCI. Now, a couple of things more to say. I'm very grateful to you young people. My two daughters, when I gave my inaugural lecture, spent the entire inaugural lecture asleep. And they only woke up when the audience applauded at the end. So, well done. Well done. And you are one of the first inauguraries here to have at least familial audience participation, if not full audience participation. But I think you can tell by the reaction of everybody that it was a great inaugural lecture. And I think, I don't know, I didn't tell you this before, but in my experience, you and I can go to rooms with a couple of thousand people in them, and we know that probably a third of them are not listening, a third of them are on their email, and there's a few that think there's something in what you're saying. But it is always far harder to stand up and give a description like this in front of family, friends, and people that you care about. So congratulations for a brilliant inaugural. Well done, Angus.